All right, so hi everyone out there in YouTube land. And we're picking up where we left off. So the topic is gene regulation. How do we control gene expression? And what we've been doing is looking at first some of the ideas behind it, the fact that we can have a single cell that has the ability to differentiate into multiple cell types, and that requires very careful control of gene expression. And went through a little diagram there earlier about the multiple steps in eukaryotic gene regulation, and there's a lot. Prokaryotic gene regulation is a good bit simpler, and so that's where we've been starting. And what we've kind of picked up on is the idea of how we can control a gene in response to different kinds of metabolites. Specifically, we've been looking at the lac operon and thinking about what we need this system to do. And specifically, because we don't want to produce the enzyme cassette of the lac Z, lac A, and lac Y. These are the three different parts of the ability to break down lactose. If we don't have lactose present, well, that's a pretty easy one. Now, the more interesting side of this is if lactose is present but glucose is present, the bacteria would prefer to consume glucose. And if we have glucose absent and lactose present, how are we able to operate that? And it's a fairly straightforward thing. So we've got these two different kinds of control theories or control ideas. We have some kind of binding protein, in this case, the lac repressor. So the lac repressor it's going to block the ability of RNA polymerase to move through in it +1. So the lac repressor blocks the progress of the alpha 2, beta, beta prime, sigma from both binding and initiating transcription. All right, so that's a fairly easy, at least in my mind, concept. Right? We got this big old thing hitting there in the middle of the road. Now, what do we do if glucose is present and lactose is present? How do we work that angle? Well, it turns out that for that side of the equation, we've got a -35 sequence that is not ideal. And so, in addition to sigma factor, we need another factor to help the RNA polymerase bind here. And that is going to play into the role for glucose metabolism. So, let's imagine then that we've got lactose present, so the repressor is gone, but we have plenty of glucose. So we're eating our glucose, enjoying ourselves, but glucose begins to go down. It goes down, it goes down. It hits a critical level, and the level of energy coming in hits a critical spot. What do? We look up and go, uh-oh. You've been eating all this wonderful glucose, and now it's gone. What's next? Well, it turns out that when you reach a certain critical threshold of metabolite, whatever the carbon source is, that below that, so if we have, let's just go ahead and say, no glucose, because we ate it all, the cell goes into stress, into a stress response. And one of the things that is produced from this stress response is a modified molecule based on adenosine triphosphate. We have an adenosine that has lost two of the phosphates, so it is a monophosphate, and it actually undergoes a cyclization, and the structure is interesting but not all that critical. It generates a molecule that is known in the business as cyclic AMP, abbreviated, of course, as cyclic AMP. Cyclic AMP is a critical alarm signal. When cyclic AMP starts getting generated, that's the bacteria's way of saying, hey, we're running out of metabolite, do something fast. We're going to start seeing in a few weeks when we get into cell signaling, cyclic AMP is a critical molecule in eukaryotic systems as well. Like when you get a blast of adrenaline, you're actually responding to cyclic AMP. Well, cyclic AMP as the stress system signal, when it goes up, it binds to a protein that is known by a couple of different terms. It binds to a protein called catabolite activator protein. And it's also known as catabolite repressor protein because it can do different things. Regardless, this protein that we will call CAP. What CAP does is it binds to the less perfect minus 35 side, actually kind of around it. So CAP is going to bind actually here. We got the CAP site. And so yeah, I'm going to erase or change that arrow up. CAP binds here. And what CAP does is it allows for RNA polymerase to bind to this less optimal minus 35 site. So we have the machinery now in place. In that if CAP is present, this minus 35 site now starts to actually work fairly well. So let's see, do I have my room over here? Sure. If CAP is present, then the RNA polymerase holoenzyme it can now bind to the minus 35 site. So that's taking care of, can you see that? Yeah. That's taking care of the problem. And now, if there's no lactose present, the lac repressor is still bound, and you're not going to make any progress. But if lactose is present, then this repressor binds to lactose, it diffuses away, it loses its binding affinity, and zing zao, you now begin producing the large amounts of this RNA. So this is how we can have this very fine-tuned system of different metabolites, glucose being present or absent, and I'm just looking to make sure I haven't forgotten anything. There you go. So we have the CAP binding site up here. We have the RNA polymerase. Actually, as I'm looking at this red box is supposed to be my CAP binding site. Worry not about that. We got the minus 35 site that's not great. We got the minus 10 site that's doing just fine, but it's being blocked by the lac repressor. And then we have the CAP site that is required. So that's how we can actually modulate this thing in both directions. And bacteria and different gene regulation bacteria use all kinds of different models. I mean, there's repression, there's activation, there's negative control and positive control. And it's all based on these two basic kinds of models, more or less. There's a couple of other interesting things that in molecular genetics we talk about, but for now, this is pretty good. You either have a poor minus 35 site and you need an ancillary protein, or you've got some kind of blocking system here, and this is responding to some kind of molecule, and it can work in a couple of different directions. Like for a feedback mechanism, say if you're producing an amino acid, when you have a certain amount of amino acid comes up, so you reach a certain threshold, that amino acid can then bind to a repressor protein. It binds, and now that repressor protein lands there and stops the whole machine. And then when the amino acid level gets sufficiently low, that amino acid now diffuses away, the blocking protein is now no longer binding, and polymerase can occur. So using these different models, we actually can control gene regulation, and you can build either actual or theoretical models of gene regulation, and they work really quite well. And it's one of the interesting things I think about in terms of linking together the idea of Km and binding affinity and the sort of thermostatic regulation of this thing. You know, I think we've talked about this before, but when you have a thermostat, you have a set point. And, you know, the way the thermostat works, particularly with the air conditioning system, but probably with furnaces as well, is there's an offset. So you set your thermostat for 68 degrees, and it's wintertime. At 67, the thermostat comes on and warms the house up. It goes past 68, it overshoots, and goes up to 69, and then the thermostat kicks off. Because if you tried to keep it right at 68, you'd have that furnace kicking off and on a whole lot, you know, just all the time, and that's not good for the motor and doesn't work very well. And the same thing with air conditioners. In fact, you know, with the air conditioner compressor, there's a certain amount of shutdown time that you have to let things, all the fluids settle out before you can turn it back on again. So you have to have this offset. And the same kind of thing is happening, and this is now depending on the Km of these different proteins. So the Km, our binding affinity, just as an example of the lac operon, with the lac repressor, how much affinity does it have for lactose? Does it bind lactose at real low concentrations and then diffuse off when you have just a very small amount? Or does it take a fair amount of this material present before the binding site is now actually going to bind well enough to cause this thing to come off? And so by changing individual amino acids, we actually can alter the Km, and in a sense, we can alter the offset of the thermostat. That is, you can make it either way more sensitive or way less sensitive. And that can have some interesting results. Now, the only reason I say this is we start talking about the eukaryotic systems. We actually can see that very slight changes in a single amino acid or a single nucleotide on the binding site can result in slightly different sort of binding affinities, and this can result in slightly different amounts of a gene being turned off and on. And this is probably what is involved in part in the development of the face. And so the degree to which your cheekbones grow is going to be in part responding to differences in binding affinity of these different kinds of molecules. And so I always think it's a pretty cool notion. We're getting better at it. And I've been, you know, standing up here thinking about this for a long time. Like in the movie Gattaca, if you've ever seen this, another one of the prerequisites for the class. You take a drop of blood, and from that drop of blood, they extract the genomic DNA, sequence the genomic DNA, you know, instantly. And using your DNA sequence, you can actually put up a picture of what the person looks like. And it's based on the slight differences in both the amino acid sequence of DNA binding proteins and the slight differences in the nucleotide sequence of the sites where these DNA binding proteins bind. It's just a thing to think about. And so we really are getting closer and closer to being able to actually develop a face based on DNA. And this is actually something I saw, I bet close on to 15 years ago. An artist, biochemist in New York, was going around and plucking random bits of chewing gum and hair and just things from New York, sequencing or amplifying up discrete portions of the genome, sequencing them, and then she had a fairly primitive modeling system where if you had these specific sequences, you might have, I mean, like things like we know, like blue eyes and curly hair and skin color are actually pretty easy to figure out. Facial characteristics are a little bit more difficult. It's not single genes, it's a couple of different things kind of coming together. But she's able to make a guess at what a person's face would look like. And I mean, she has a model of, like she was doing 3D printed masks, and she had a model of her DNA sequence face. And you look at it and it's like, sisters, I don't know, with appropriate makeup maybe, I don't know. But you do get some really interesting things. And it's a new kind of another wave in forensics, the ability to take DNA, and while you may not be able to actually have a CODIS match, somebody's in the database, you can begin to say, okay, this is a person of, most likely Irish descent, and they've got this color hair, this color eyes, unless they've actually done some sort of alterations to themselves. So, that's why I, of course, always wear contact lenses of a different color, and I've got, DNA sprayed around me so I can always spoof the forensics technicians. But that's for a different story. All right, so now, eukaryotic transcription regulation is going to be a different game. I mean, not surprisingly, the prokaryotic model just doesn't really fit in a number of ways for the eukaryotic system. One thing is you don't have a single spot where you can get a bunch of RNA that will produce 5 different proteins or something. Second, we know that RNA polymerase doesn't have intrinsic binding affinity to the promoter. So we've got some things we can kind of work around there. And a couple of models come out from here. So we're going to kind of shift now from the prokaryotic system to the eukaryotic system. And there are a lot of regulatory systems that we're not going to fully talk about, but a couple of biggies we'll get. All right, so a standard way that we can begin regulating transcription in eukaryotes. So let's just go ahead and title this up. And this is, a title for an entire class in graduate school, and that would be eukaryotic gene regulation. And we got a couple of points here. So we know that blocking the RNA polymerase from proceeding, you could do that. There's really not a lot of good examples of that, partly because there's so many other mechanisms that we can have in play, and that is RNA polymerase just doesn't bind to DNA. RNA polymerase binds to all these ancillary transcription factors, these basal transcription apparatus factors. So what we often see then is a bit more of a complex control system in that we have +1 here. We got the -10, -35 sequences here, the upstream control regions. These are the binding sites for the ancillary factors, TF2D, TF2E, TATA binding protein, stuff like that. And then we're going to have upstream some kind of, and a couple of different generic terms, often what is known as an upstream regulatory sequence. And in fact, a classic model system in yeast is a DNA sequence that is known as a UAS, the upstream activation sequence, because when you put it there, it activates gene expression. So just if you ever see this, and again in molecular genetics, we might start playing with some UAS stuff as well. The upstream activation sequence, the UAS. And one of the things we start thinking about both these systems is it's a two-part thing. You got the DNA site, the sequence, and then you have the transcription factor. And the term that we talk about, or way of thinking about this, kind of a throwback to organic chemistry, a little cis and trans, we would say that The upstream regulatory sequences, or a UAS, regardless, we say the DNA sequence is cis-acting, and it binds trans-acting factors. So trans-acting factors. So it's a two-part system. You got the DNA binding site, which has really got to be somewhere near the gene on the same chromosome. We have the transcription factors, these trans-acting factors. These are proteins, and they can be produced anywhere in the genome, and so they are diffusible within the nucleus. That's another way of thinking about this. So, generic terms then for the upstream regulatory sequences, a UAS is one of these things. These are collectively known as enhancers. That is the name we give to the DNA binding site, an enhancer. And an enhancer, actually these were discovered, I think initially, by looking at the way viruses work, is that many viruses contain, they have the gene set that they need for replicating themselves. They have some kind of basal promoter, and then they often will have a sequence not that far away that is a phenomenal amplifier of gene expression. Because one of the things, not to get too far off track, but if you think about a mechanism of a virus, it lands in a cell, and it's got to be ready to roll right away. It wouldn't do to have a cell or have a virus that requires a specific kind of differentiation factor, maybe, or some kind of protein binding factor to actually turn its expression on. This virus lands, and it needs to be able to control its own gene expression. So it turns out that a lot of viruses have very potent enhancers at their 5' end. And so when they land, they're able to bind transcription factors that are present in the cell and just start cranking, or they'll produce their own transcription factor that'll bind there. And so the point of this is, historically, people began identifying sequences in viruses that would phenomenally amplify up gene expression, like by tenfold. I mean, it was like a magic little trick there, that this DNA sequence would actually amplify gene expression. And we'll be getting into cancer mechanisms and things, and one of the things that can happen is that the virus can land near a gene involved in cell signaling, involved in triggering cell division, and the enhancer from the virus can actually kind of feed into this other gene and cause it to be overamplified. And that's one of the kind of cancer-causing mechanisms that viruses can do in some organisms, where we call transforming viruses, and we'll talk about this later. But, all right, so we have enhancers. Now, with some of these initial experiments being done with viruses, one of the things that was observed is a set of conditions and differences that distinguish enhancers from plain old promoters. So if we were to think about sort of a test to say, okay, we got a DNA sequence. It binds transcription factors, and it activates gene expression. Well, that sounds like a promoter. Enhancers are not promoters because of the following. Promoters, by their definition, are organized right next to +1. I mean, the other side of this is the promoter defines +1. -10 is called -10 because 10 nucleotides later is where you start transcription. So the promoter is very much distance dependent. So this is, just as make our list better, a promoter. It's distance dependent. If you were to move this promoter back this way, well, -10 becomes over here. So the promoter is a very specific kind of sequence that says you will start transcription 10 nucleotides from X. Okay? I'll start. So the promoter is very much distance dependent, i.e., it defines where +1 will be. The other thing about a promoter it is orientation dependent. Because not only is the promoter saying you were to start transcription 10 nucleotides from here, it's also saying, I'm -10, that's -35, i.e., you're going that way. Because if you were to take the promoter and flip it over, you got -35 here, -10 here, you're going that way. And so the promoter is determining the specific direction of RNA polymerization. Consequently, it's orientation dependent as well. So if you flip it around, start transcribing the other direction. Some of this stuff actually comes about from the way we do experiments with DNA and the way enhancers were first identified. I get very excited about transcription factors. So what people were doing with basic recombinant DNA techniques is you set up some kind of marker gene. Green fluorescent protein back in this period hadn't been identified yet, but it's a great one. And you have a basal promoter, and you look to see, all right, if I put this DNA sequence in, do I activate transcription? Do I get the bacteria or whatever, or the cells start turning green? Now, what happens if I take this piece of DNA and flip it around? Well, with a promoter, if you flip it around, you're running the other direction, you don't get anything being produced, and that's why we say this is distance dependent and orientation dependent. The distinct difference is with an enhancer, they are by definition distance independent, meaning they can work from, sometimes as far away as 50,000 nucleotides, and I imagine if you start digging around, you can probably find some that work even further away than that. 50 kb is a pretty good spot. If you're looking for an enhancer and you got to go out further than 50,000 nucleotides to find something, you probably should start looking at a different gene, or figure a different gene out for your PhD work. Additionally, they are orientation independent. Meaning that they don't have to have a specific 5', 3' direction. They can be this way or that way. And this is because they are simply a binding site for a transcriptional activator. There are silencers as well, but they're not as common, in part because the gene, unless you do something to it, it's silent anyway. So you can find articles and experiments that identify some kind of silencing thing, but I'm not entirely sure that's not artifact. Regardless, enhancers are really good at binding transcription factors. These transcription factors are in turn transcriptional activators. And this is the best model for how they work. And I could put the picture up, but we'll do a little drawing. Why not? So, in order for an enhancer to actually work, what it's doing, and this is sort of the best model, and we got good space. Here is +1. Here is -10, here's -35, and this is where all of the different kinds of transcription factor, TF2Es, TF2Ds are binding, RNA polymerase is binding here. We got all kinds of stuff setting up. An enhancer works like this. It is actually going to cause the DNA to loop around in that here is the enhancer, And here is the binding protein that is the transcription factor. And it is stabilizing the construction of this little nest for the RNA polymerase. And so without this kind of twist around and little pocket, transcription doesn't work all that well. And so in order to get good eukaryotic transcription, you probably have to have some kind of, you have to have the basal promoter, but you probably also have to have some kind of transcriptional activator system as well. So another kind of gene regulatory system that actually is, I think, an interesting game when you're trying to do techniques where you're adding genes to a recombinant, making a recombinant eukaryotic organism. And a perfect example is genetically modified plants. When you're doing genetic modification to a plant, what you're going to do is take the gene that will confer resistance to the herbicide, or take a gene that will generate a poison for insects or whatever you want to try and do, and you blast that into the genome. The integration of this piece of DNA is pretty random, and it's not at all unlikely that the DNA will land at a spot that is kind of transcriptionally silent. In genetics, potentially, you talked about histones, which are a set of very positively charged, very basic proteins, and the chromosome wraps around the histones, almost like thread wrapping around a spool, and that's how you begin to condense chromosomes, and we're not going to go into it, but regardless, if you get into a spot that is highly condensed, your gene may not be activated and may not get turned on very well. And that's another mechanism by which genes can be regulated in naturally occurring systems, is there are genes that, depending on the differentiation state, they can be tied up in chromatin and actually being condensed into what's known as a heterochromatic region, a very condensed, non-translatable or non-transcribable spot of the genome. And if you land your recombinant DNA molecule there, well, it probably won't work very well, and that's why you probably would do a bunch of different individual landing spots just to find one that works better. The technology known as CRISPR-Cas9 actually allows you to direct specifically at what portion of the genome you want to put something in. And so that's actually a way now to get around that, is you can direct specifically. Okay. Now, let's throw, actually, I'm going to throw up a couple of pictures here. There's a couple of cool images to look at. Get the 8. Yeah. So the game then becomes this two-part story. You got to have the transcription factor, and you have to have the transcription factor binding site, or the enhancer. And a key mechanism then behind gene regulation is turning on specific transcription factors. And we can go on for a while to tell you about transcription factors that will convert a basic fibroblast into a muscle cell. One of the cool things is it turns out muscle cells are not that hard to generate. Trying to find transcription factors that will differentiate a cell into a neuron is a pretty cool trick. Differentiating into all these different kinds of cell types, that's part of the sort of the next wave of what biochemists are figuring out. And it's not all transcription factors all the time. I think that if you'd asked me, 30 years ago, I would say, yep, we're going to see transcription factors. That's all you need to know. If you can control transcription factors, you can control gene expression. And we now have a lot more understanding that it's also cell signaling and hormones and receptors that are in turn interacting with transcription factors. But in the fly system, you can so carefully tune gene expression that fairly easily you can develop a gene construct in which you will have a single cell along a whole band, like one of the segments of the fly larva. A single cell, you can light up that one cell with a new gene. And so the precision by which you can direct gene expression using transcription factors is pretty staggering. We're going to look at just a couple of pictures here to see how complicated it can be. So this is the overall model. We have the transcription factor, and then there's some kind of mediator guy that is establishing a happy nest for all of the basal factors and RNA polymerase. And that's how you begin transcribing at a high level. So this was the other model in which you have the histones binding the DNA up and making a region of chromatin that just isn't accessible to RNA polymerase. Oh, that's kind of boring. But this is now a good bit more complicated model, so it's probably how things really do work, particularly if you have one cell or a specific set of cells that you need to have a gene being turned on. You're going to need more than one transcription factor. And so these kinds of models begin to develop in that you have multiple regulatory sequences. And you can do all kinds of interesting combinatorial models where if you've got this one and this one and this one, you'll have very low levels. If you've got this one, you have much higher levels. And then, lots of different ways of fine-tuning this thing. And it generates something like this. So this is the molecular mechanism for how a gene called even-skipped is regulated. And we won't go into fly embryology, although even if you just ask me a little bit, I'd jump into it in a second. Flies hatch out as larvae. They're holometabolists. They go through complete metamorphosis. And a larvae is just a fancy, or is a sort of derogatory term for caterpillar. Caterpillar is a lovely term, but they're all maggots. And so you've got the larva is segmented. And not to belabor the point, but you have these multiple segments that then correspond to different body parts and things like this. All right, so the adult actually is carried along as little packets of cells, and the adult has a whole different mechanism by which it differentiates. But the larva hatches out really fast. After, like with flies, at 24 degrees, they go from a freshly fertilized egg, now oocyte, or now zygote, to a hatched out larva in less than 24 hours. I mean, that's like really cranking. And in early embryogenesis, the patterning is beginning to establish the individual segments. So this is what even skip looks like if you take an antibody and stain an embryo, and you see these stripes. Do we remember fruit stripe gum? Nobody knows what fruit stripe gum. I don't think they make it anymore. It's the zebra with the stripes. So I used to do antibody stainings, and you had this little dish filled with stripy embryos, just like, oh my God, it's the fruit stripe zebra. And what you can do is begin taking apart the regulatory sequences and actually identify down to individual spots what is controlling the expression at one spot. That's a pretty cool experiment. And so what we got here then is a reporter gene where they take the LAC-Z gene from bacteria just because it's really tough and there's a lot of staining systems we can use. GFP would work, but you need a fluorescent microscope. And you've got the Tata box, you've got the various sort of basal promoter stuff. They're not showing minus 35, but it's got to be there. And you can identify a specific regulatory element they call strike 2. And this is binding a couple of different transcription factors, and that's what establishes the expression right there. Now, the next question, of course, is how do you establish the transcription factors? I mean, it's kind of like who cuts the barber's hair, or I don't know, whatever. And that is a whole other game. This depends in the fly embryo. It depends on a whole set of other transcription factors that have been packed into the oocyte by mom, and she fills the embryo starting at the back end and begins filling it up to the front end. And so there are some RNAs and proteins that are expressed back here that then taper off in their levels to the front. And then there's some guys that are not expressed at all until you get to the end, and then they're expressed at real high levels. And this setup allows for a gradient of transcription factors that gives you this. It's a pretty cool little system. And as it turns out, placental embryos, like you and me, and for that matter, most animal embryos, or all vertebrate embryos, are developing in a good bit more complex way. And so this model doesn't quite work for human embryos, but still. This is what it's looking like, in that little chunk actually has binding sites for a whole host of other transcription factors. So, the fact is, even skipped is itself a transcription factor. And it's got all these different kinds of transcription factors binding. We have hunchback and crouple and bicoid and giant, bananas, carrots, apples, and a blue mushroom, all combine in concert to create gene expression at that stripe. And so if you don't have everybody set up, it won't work. And it's a pretty amazing model in that it allows for a whole range of gene expression games. You start looking at these different transcription factor systems, and you can begin to build a construct that will express under very specific conditions, at very specific times, at very specific locales. Really, we can begin to build a genome that will express genes in different tissue types at different times. This is how it works. And so there are a whole range of different kinds of models. So here is an example of a system that is using steroid hormones. And I can actually draw this from better. What we're looking at here, I don't know what we're looking at, but regardless, a cluster of cells and then neurons, I know. You got these different regulatory proteins that start coming on, and this is how you differentiate, this is an example, hematopoiesis, where you have a basal, sort of a stem cell that generates the whole different types of cells that are in the blood. And, you got cell division, a regulatory protein is present in one cell but not in the other, and you begin this combinatorial generation of regulatory proteins such that we end up with multiple cell types. And so, not only is it a theoretical model, we actually can find examples of how this works in developmental biology. What are we looking here? Yeah, so this is a standard kind of system in that we take a tissue culture cell that really isn't particularly differentiated, what is known as a basic fibroblast. And you can generate fibroblasts, like by collecting cells from the inside of your cheek, you would get fibroblasts, and you start culturing them. And they last for about a week or so. They probably are good for about a week's worth of cell divisions once every day. But by adding different kinds of transcription factors, you can even actually inject them into the cell, or adding different kinds of hormones that will in turn turn on transcription factors, we can begin to induce different kinds of cell types. So this is a model system for how we begin to actually understand differentiation. Here is a weird-looking figure, and I was this close to cloning that gene. This close. This is a gene that in flies is known as eyeless. In fruit fly world, the genes are typically named after the mutation, which is kind of unfortunate. So the presence of this gene allows for the differentiation of the ommatidia of eyes. The absence of it means you have flies that have no eyes. And what a group in Germany, Walter Gering's lab, I think it was, they expressed the eyeless gene at a specific spot in the leg. I mean, it's actually the way legs form in flies is there's these little packets of discs that are known as imaginal discs. And they're being built as the larvae are crawling around in the food. So the larvae go through multiple instars. When they get ready to undergo metamorphosis, they have these imaginal discs that have been built. And the disc, like the leg disc, it looks a little bit like if you've ever seen a collapsible camping cup. That's what it looks like. It's all flattened out. And then molting hormone causes the disc to actually go whoop and expand out. And so you got the base and you got the tip. And one of the cool things about this is that it doesn't require any cell division. It's just cell movement. And so you literally have this flattened stack that then opens up into this tube. And what they did was cause the eyeless gene to be expressed in one of the little concentric parts of the tube. And lo and behold, you produce an eye here. Now, the eye wasn't innervated, and so the fly was not seeing from its knee, but it was, in fact, operating in this way to differentiate a whole range of cell differentiation types to generate the eye. I mean, you start to get pigment. There is a whole set of really cool gene interactions in which you have a central cell that says, all right, I'm it. It secretes a regulatory protein, and the cells surrounding it bind that and say, okay, you're it. We're going to be the surrounding cells. And you begin to build this octagonal structure that is the ommatidia. And that's what's happening there. The reason I was this close to cloning it was when I was a postdoc, the lab below us was a human genetics lab. And they were working on a region that specifically is involved in something called Wilms tumor, which is childhood kidney disease. And we'll actually get into Wilms tumor, the gene is responsible for it later in the semester. Wilms tumor is a childhood kidney disease, and it turned out that people that would get it would have deficiencies or deletions in a specific part of chromosome 11. And in addition to having Wilms tumor, people that had these deficiencies also would have some genital urinary defects, mental retardation, and something called aniridia, in which they actually were born without an iris. And so this was known as the WAGS syndrome, was Wilms tumor, aniridia, genital urinary, and I don't know what S was, but regardless. So there's a guy that was cloning genes in this area and trying to find the Wilms tumor gene. They had a gene that was a transcription factor. They knew that because it had the structure that bound, that looks like the kinds of proteins that bind DNA. They didn't know what it did, though. And we were thinking about, okay, what if you look for a homologue of this gene in flies? So this rotation student came down to our lab, and we were only vaguely looking around for it. The technology, I now know how to do it a lot better and a lot faster, but then it was going to be, a lot of work, and who knows if we're going to be able to find it. And so we did a little bit of screening and analysis, didn't come up with anything, and probably a year later, this is on the cover of Nature, and that this is the fly homologue, the aniridia gene. So there you go. Yet again, one of my brushes with the cover of Nature. All right, we'll pick up on Monday. And just to say to everybody in TV land, the homework assignment that I talked about, A, I'm swamped with grading and behind. B, I have it all ready to go and drop it as a file. C, if I get to it today, I will. If I don't, I'll get to it on Monday, and it'll be due in April. All right, ciao.